Hi hires, so Sustainability and Interdependence Unit. In particular, we are looking at what happens when you produce hybrids. Okay, so crossbreeding. So this has been mentioned already because we're looking at it in terms of inbreeding depression or avoiding it, and it is rather important that you keep this going to a certain extent to prevent inbreeding depression. But also it could mean that you can bring in a particular characteristic, so one that you don't have in one particular section of the population, you do have in another, bring them together, and then you can use that. Okay, so, but what you have to remember is if you've got one, we're talking about plants at this point, if you have one cultivar that's true breeding for whatever you wanted, so say it was a height thing that you now got a particular height that you wanted to true breed it for, and that's what's happening. And then you have got another cultivar that's true breeding for another thing you want. So not only height, but say you wanted a particular bushiness to the plant. Okay, so these ones are great, but the first one is the right height, but is all spindly, and the other one is all nice and bushy, but it's too short. Okay, so if you crossbreed them, what will happen now is you end up with a range of heights, and you end up with a range of bushiness to spindliness. Okay, so you've got heterozygosity in both of them now. So you're not quite back to square one because you now have all the genetics in there that you want, but what you then need to do is back cross again, select, and you can breed them back to both being true breeding in one new cultivar set. Okay, so that's one way of doing it, and it works well, but you're talking about 10 more years depending on the life cycle of the plant you're looking at. So one of the ways that you need to be able to talk about it is in terms of what you would get with that first generation of taking two cultivars. So when you take those two true breeding ones that potentially are not super fabulous in other ways but are great in the way you want in one particular thing, if you bring in two plant species, so let's just imagine a kind of thought experiment with this one. So if you've got plant species 1 and you've got 3 characteristics there, okay, gene A, gene B and gene D. Okay, so species 1 is true breeding in A, B and D. It's true breeding to the recessive in these two and to the dominant in this one. Okay, and here's species 2, so it's true breeding for dominant in the A and D, and it is recessive in B. Okay, now if you do a cross with these, okay, hopefully you can see this simple example of this. The gamete for these ones would be A big B little d, and for this one would be big A little b big D. So there is only one option of a gamete from each of these. So when I join them together, every single one of the F1 is going to be big A little a, big B little b, big D little d. Now let's assume, and it is an assumption, but let's assume that the dominant is actually the kind of most impressive of the traits in each of these. Okay, so F1 is going to have all of those dominant phenotypes. So this particular set of plants, the F1, is kind of better than both of the parents. And this term is the one that you need to know, hybrid vigour. Okay, now there are two ways you can go from here. Now, assuming that you have closely enough related ones, then you can go on to breed them from the F1 to the F2, which is what I'm going to just go on to talk about. But there is another option, which is that this is where you stop. You may end up getting an F1, but at the F1 they are too different, so you end up with not breeding further on. Okay, so that would not truly be about breeding different plants, it would be producing one F1 and then that's you done. So effectively they're different enough that they're acting as different species. Okay, but assuming that we have a closely enough related that they can make gametes quite comfortably, let's look at see what would happen in the F2. Right, so this is going to get a little bit more complicated, even though we're only looking, and this is important, think about this in kind of the fact that we're only looking at something very, very simple, although it might not feel like it at the moment. We're only looking at 3 genes, A, B and D, and we're only looking at the potential for 2 different alleles in each of these. This is not the reality a lot of the time, okay? So as you keep going on, if you take the F1 to the F2, you start getting something much more complicated. So it's not true breeding because we've got more and more of a mix. So here we go. You might need to do some pausing of this. Try and work out all the possible gametes from this F1 that you have. Okay, you should have 8. Okay, here we go. These are your potential gametes. Okay, so we could have big A, big B, big D, could have big A, little b, little d, and so on and so on. Okay, so here are our potential gametes from these 3 ones that you have here. Right, Punnett square. Now I know the Punnett squares you have always done were the simplest possible ones because you just had a little kind of 3 by 3 because you only had say 2 potential ones like that. Okay, try and draw out Punnett square, 9 by 9 grid. Put your gametes, enough space for you to be able to fill them in. Now the reason I'm asking you to have a shot of this is just so you can see where it's going, but obviously I'm going to show you. Okay, fill in everything, and finally, just to make you feel better and get to do some colouring, colour code. Now, so what you're looking for is colour code if you've got all 3 dominants showing, so any combination which actually has a big A, a big B and a big D. Okay, colour code for 2 dominants. Now I'm not asking you to justify between the 2, so just any 2. So if you have a big A and a big B, or a big B and a big D, or a big A and a big D, that kind of thing. And if you've just got 1 dominant, so a big A or a big B or a big D. Okay, so what I'm trying to get you to see here is how much variation you can get between an F1 where everything was the same to an F2. Okay, so this is what it's going to look like. Now I've colour coded in red for anyone where you have all 3 of them showing dominant. Blues are where you have 2 dominants, and greens are where you have 1 dominant. And one of these you will see in the middle here, but here, where we've actually got all 3 recessives. Okay, so say you wanted to keep all 3 dominants. You only actually have up here 1 true breeding individual, which would be your best thing to use. Okay, but then you have all of these guys down here, and all of them are red, so over here, up here, over here, and all the way along here, which are all showing the 3 dominants, but some of them have 1 recessive in here in the A. So what would happen is that these traits would breed true, but this one would throw up the recessive every so often. Okay, this one would breed true for the A and B, but it would throw up the recessive for D. And then you've also got some of the blue ones that I've got in here. We have 2 dominants that are showing up, but again, I haven't split these up. So these are A and D, A and D, A and D. This is an A and B one. Okay, so this one we'd end up true breeding for A, true breeding for B, but in the recessive, and is going to throw up combinations on the D. So this was just 1 generation away from everything being the same. Okay, so this is an interesting issue. Okay, so if you want to get everything from those parental strains, that's what we're meaning about backcrossing, selecting, or your other option is just keep the 2 original strains and keep producing that F1. Okay, but what also might be, just to remind you, is that you might get a new source for selection and breeding because you brought in different variation. Okay, and that's that.